All right, so I think I got cut off at the end of that last video. So the last part here, we were talking about the different functions of media, and the last one was an enumeration media. So enumeration media is used to determine basically the number of bacteria, or it could be fungi within a sample. And this is used by the food and drug industry to test food items for the allowable number of bacteria that is within that product. So there's only a certain number of microorganisms that are allowed in milk or certain foods that can be sold here in the United States. So enumeration media would be used for that. Now let's move on to our next I. So we have thoroughly covered inoculation. So we talked about the 5 I's to begin with. So we had inoculation, we talked about the different media that can be used, and now we want to move on to incubation. So the culture, once it is created, is placed in an incubator that helps to keep a temperature, keep the culture at a certain temperature, and can also control the levels of gases that are present. So an incubator can be small enough that it fits on top of a bench in the lab, or it can be standalone, or it could be even the size of a room. The incubator basically helps to keep the temperature constant. The temperature can be adjusted based upon the microorganism that you're culturing. Usually the standard is 37 degrees centigrade if it's any microorganism that you're trying to grow that has been isolated from the body, the human body. So they grow best at 37 degrees centigrade. So the culture is placed in the incubator for a certain amount of time, usually about 24 hours, to allow the bacteria to replicate. And once it starts to grow to a certain size, you'll be able to see a colony. So typically the culture is kept in the incubator until you see colonies that are visible on a plate, on a solid media, or your broth becomes cloudy, then you know that the bacteria have multiplied. After incubation is isolation. So this is the picking of a colony from a solid media that is typically then transferred to another media. But let's take a moment here to just kind of review our definition of a colony. What do we mean by colony? So if we think about it, right, we have selected a specimen. So here we've got a broth culture that is cloudy looking. We know there's microorganisms growing on it. And now we want to plate out those microorganisms on a solid media. So we might take what we call a loopful of that broth. So we can use what's called an inoculating loop, which is basically kind of a piece of wire that has a little loop at the end that is of a standard size, and you dip it into the broth and it will then create kind of like a little film across that loop, kind of like when you're dipping a bubble wand into a bubble solution, you see the film that's on that bubble wand. So we take that little bit of measured amount and we will then streak it out onto that solid plate. When you streak it out, you don't see the bacteria at all, but you know that you have placed it onto that solid media. So in the top here, we see we've got a mixed sample of rods and spherical bacteria that have been selected. And once we place it onto that solid media and place it into the incubator and give it the right environment, the right temperature, it now has the nutrients within the media so that it can start to replicate and grow. The bacteria are going to start to divide and keep on dividing so that we have many more of them. So you see a lot more of these white spheres and a lot more of these red rods. And as they start to grow and divide, and there's more and more of them, they go from being something that is, microscopic to something that is then macroscopic. So before we could not see them when we originally streaked them out on the plate, but now that there's so many of them and they're growing on top of each other, that we can now see them on the solid media. And so that mass of cells that have divided and divided is called a colony. And all of those cells came from one parent cell. So we can think of all of the cells within that colony as having the same genetic information because they are all derivatives of that original cell. Same thing with our red rods. They all came from that one cell that went through multiple cell divisions. It's asexual reproduction, so they should all be exactly the same. So all members of that colony should be alike. So when we isolate a colony, we are selecting a little bit of that colony, and then we typically will streak it out on another plate. So that way, and we go from a mixed colony to having just a single microorganism growing, or a pure culture. Our last two I's are inspection and identification. And we're going to just kind of briefly touch upon this, and we'll discuss these two in further lectures. So mainly we're looking at the appearance of the colony with inspection. We're looking at the shape of the colony. We're looking at the margins of the colony. What's the texture of the colony? We could also be looking at how it reacts to the nutrients that are present in the media, or the different chemicals that might be present in the media. How does it utilize it? Is it able to ferment it? So we're doing all of that during the inspection phase. Identification is basically naming the species that is present in the culture. And to do this, we may use certain immunological or rapid tests, as you would see in a doctor's office, to help confirm the ID of the specimen. So we have completely covered all of the I's. We've talked about inoculation, incubation, isolation, inspection, and identification.